Hello everybody, I am Dr. Farhan Zamir, an adjunct professor and academic specialist at Biotechnica, Bangalore. Today, we would have a very special discussion on a crazy topic, and the caption for today's discussion is Synthesis of Artificial Blood. To know about this, what we'll try to do is we will dwell into the topic and then we will see how best we can synthesize blood. Let's dive in. So when we start with a discussion like this as synthesis of artificial blood, which is not natural. Now, just to give you a brief background of what is blood, okay, blood is the fluid of life. Okay, especially in humans, it is fluid of life. Now, any point of time, if there is any variation like depletion of blood or there's a loss of blood, and this can lead into fatalities. Now, for today's topic, we would thank one of our viewer and one of our student who said in his comments saying that, sir, can we actually synthesize an artificial blood? Now, that gave us a thought of designing this entire video. Now, to start with, the concept of blood, so what we will try to do is we will look into the agenda, because we need to have a proper flow, and with the agenda what we will try to do is we will try to first understand the history and discovery of blood, how exactly blood was actually discovered, and we will also try to understand the blood and its components, and then what is the necessity for actually synthesizing artificial blood. If that is the case, then what are the various methods and methodologies in which this blood could be synthesized? What is the advantage of me having blood as one of the major component as into my synthesis? Then what are the limitations? And then finally, we will try to say what are the various scientific conclusions which can happen with the synthesis of blood. Now, let us try to go on to our first slide. To have a preamble, so when life started, everybody had their own concepts. Many a times science was very godly, or it has been given, or it was been devoted only for a special category of people which were been connected to gods. Okay, so maybe we can call them godsman or a messenger or something like this. But apart from all the theological relationships, okay, what we will try to do is we will look up life in the angle or in the light of biology or in light of science. So when we look into the entire evolution of body or a given cell, the few misconceptions or the first primary concepts were the entire life started with earth. This was combined with fire. This was later combined with air. And finally, we had water. So to make up life, these were the four major constituents or the ingredients which were the primary foundation for the evolution of life. So it was earth, it was water. You can still take earth as soil. So it can be earth, water, air, then fire. Now this was again further classified into dry and moist, hot and cold. So with these four combinations, people believed that this is how the organism or life existed on the face of this earth. Now later, this combination of dry, moist, hot and cold led into certain biological fluids, and these biological fluids were called as blood. It was called as phlegm. Called later, another variety was called as the black bile, and the other variety was called as the yellow bile. Now, there are a lot of misconceptions, lot of old theories which were true at one point of time. At this point of time, they have been absurd. Now, what we need to just understand is science did not evolve overnight. So it was not a miracle. Now, with a lot of efforts, lot of thought process which has gone into it, and then the emergence of understanding of life has been evolved. So let's see how exactly the concept of blood and the circulation of blood evolved. A great thanks to a person called as Galen. Okay, Galen was a polymath. He belongs to the older part of Greece. He was a polymath. He was a physician, a surgeon, and very importantly, a universal scholar. Now, Galen, he had his own ideologies. I would like to bring Galen into our discussion because it was his theory which was being believed to be true for more than 1200 years. Now, when a theory has been believed by a group of people on the entire face of earth for 1200 years, that means to say there should be some reality, there should be some strength, okay, some basis for his theory. And because of this reason, even in under modern science, Galen has been mentioned as the father of systematic medicine. Now, the Galen's hypothesis, to understand Galen's hypothesis, we need to dwell a bit more. Galen had his own ideas, and then he said there are certain passages, there are certain pores. In these pores, the blood is actually entering and air is also entering with the blood, and he actually gave up a concept of an open circulation system. Now, what exactly is an open circulation system? We will try to dwell upon when we go into the next slide. So remember, one of the major hero of blood circulation in humans, the concepts were given by the concept was given by Galen. Now, Galen, after some point of time, was actually disproved by a great physicist. His name is called as bin al-Nafis. This scholar, he came up from 13th century, wherein the concept of pulmonary microcirculation was being given by Nafis, and then until today, Nafis is being called as the father of circulatory physiology. Okay, a great scholar who has given a lot of concepts, not just in blood, but however in various other human mechanisms and physiology. Now the entire story, changed. It took a paradigm shift when a great scientist called as William Harvey, Sir Dr. William Harvey, okay, he came up with a concept of completely remolding the concept of blood circulation. I would urge you people, ask you very humbly to go read this book, which is called as Great Minds of Science, William Harvey, Discover of How Blood Circulates. Excellent book. Okay, this video also gave me an opportunity to read this book from Lisa. Extraordinarily illustrated. Now, to talk about William Harvey was the first person who gave up the concept of blood circulation. He was born in England in 1578 and died in London in 1657, and he got educated from University of Cambridge and University of Padua. Now, until now, we were with Galen's hypothesis, and a part of Nafis gave us another concept of pulmonary circulation, but William Harvey proposed for the first time a newer mechanism of blood circulation in humans, and this system was called as closed circulatory system. So, to make you understand this in a much better way, look at this figure. Now, William Harvey did a very simple experiment. The experiment talks about the ligator. So what he did was he took up a hand and then he ligated it, okay, with a band which was used in two different compressions. One, he was completely compressing it, and the second one was which was with a loosen mechanism. Now, when he was actually compressed the entire ligator, the ligator was able to block both arteries and veins. Both arteries and veins were actually blocked. And when you look into that kind of a mechanism wherein both arteries and veins have been blocked, so what would happen? There is no pulse. When both arteries and veins have been blocked, especially your arteries, when you block your arteries, that you don't find pulse. But however, under this condition, the entire hand was numb. It was actually cold. So this gave an idea to William Harvey that it is not of an open circulatory system, it is somewhere, it is connected and interconnected between an artery and a vein. And hence, later what he was trying to do was, he was very systematically blocking the trafficking of blood between an artery and vein, and you can see the finger movement, okay, between the artery and vein, wherein if you read the actual manuscript from Harvey, okay, this is fantastically illustrated, wherein he gives up a point as A point, B point, C point, like that. It will go into N, M, L points and all these things. So the basic function, the basic, the findings of this particular study was the blood does not go as an open system. The blood is interconnected to its organ and it is into a closed circulation. This was for the first time given by William Harvey. Let me try to explain you the entire components or the 4 models which are being illustrated here. Now the 4 models are, the proposed theory of circulation. But remember, from model number 1 to model number 3, it was an open system which was being agreed, and only with William Harvey we had a closed system of proposal. Now, what is an open system? If you look into the entire component, you have the brain, then you have the lungs, then the heart, then you have the liver, then the intestine. So here, it was been believed that the air and the water exchange would have taken place because of certain imaginary pores, and these pores were responsible to transfer blood from the heart to the other parts of the body, and from the other parts of the body again back into heart was coming back through these particular pores. Okay, at some point of time you would laugh at this particular theory, but however, this is how evolution of blood circulation happened. Now, this was been believed for a very longer time until when William Harvey said that, okay, this system actually exists as a closed system and not an open system. Now, when you look into it, you can always say that your brain is connected into lungs, lung is connected into heart, heart is connected into liver, and liver is connected into the intestine. So here, William Harvey for the first time gave up a concept of the blood actually circulates in the arteries, but it is not in an open system, but however, this is in a closed system. So let us try to understand the history of the blood and how exactly blood came into the entire path of discovery. Now, in 1900, it was Karl Landsteiner, an Australian scientist, who discovered the human blood group, and he was also able to very efficiently classify them as A, B, O type of blood grouping, and then for which he received a Nobel Prize in 1930. Now, in 1903, it was George Washington who gave up an idea of the usability of blood in surgery, because until then there were a lot of people who were just suffering with blood loss, and many times they faced death because of this blood loss during surgery, and hence it was the George Washington who as a clinician or the surgeon for the first time made the usability of blood transfusion. Again in 1940, Karl Landsteiner and Alex, his PhD student, they came together and gave up a new concept, and this new concept was on the rhesus monkey factor. Okay, where now we call it as an RH factor. This was actually discovered in 1940, and in 1961, it was recognized that the platelet concentrate can actually reduce the mortality of hemorrhage under cancer patients. So, cancer patient and platelet therapy started in 1961. In 1993, for the first time, guidelines on medical assessment of for the blood donors was being published, and then in 2011, it was WHO who actually gave up certain safety guidelines in a global situation of how exactly blood donation and transfusion should happen. So the entire pathway, if you look into the most of the resolutions with the blood has happened somewhere in 2011. So when I talk about this, we are not talking about 1 century back research. It is just the last 1 decade which is now evident for having a lot of concepts on blood and blood therapy. Now, to understand what is artificial blood, I need to understand what exactly is natural blood. Now, when I talk about natural blood, you have the hematopoietic stem cells which are responsible for producing an array and spectrum of various kinds of cells. And these array or spectrum of cells, which include the platelets, the erythrocytes, basophils, eosinophils, neutrophils, monocytes, dendritic cells, B cell, then lymphoidal cells, and then T cells. These will actually create a consortium of the hematocrit, which will have both the liquid component and the pelleted component, what we call it as the serum and the plasma, or in short, it is called as the liquid component is called as the plasma, and the concentrate is called as the hematocrit. Now, we are into, we Indians, we are with Bollywood, and Bollywood is always with cinema. Now, when I talk about blood, there would be one major question that, sir, you spoke about everything about natural blood, but how exactly in cinema is the blood being produced, or how it is being utilized? Now, to look upon this, when we did a small amount of research, it was a concoction, it was a combination of something which is called as grand guignol recipe, which actually fakes blood, and this is called as blood revolution. So wherein as a sample, a combination of glycerine, a food colorant, cornstarch, and a few chemicals are being used so that the thickness of the blood is being maintained. And however, there's a beautiful recipe for this, and this recipe is called as Dick Smith's modified recipe. And in Dick Smith's modified recipe, he uses this ferric chloride as one of the oxidizing agent, so that after some point of time, ferric chloride gets oxidized from its typical brownish color to a bit dark browner color, which actually resembles the splint blood. So this is how in movies, artificial bloods are being utilized, but at no point of time, the original blood has been spent in a movie. So until here, there was a fun part wherein we were enjoying the cinema, how exactly artificial blood is being used in cinema or any kind of a movie. That was the discussion. Now, the serious part of it. Let us imagine, this should not happen to anybody, but however, let us imagine that one of our closest person who is very close to our heart, if they meet with an accident. Now, the condition is, why do they die? They die because of two major reasons. One, because of the physical damage, and the second one is mainly because of the blood loss. Now, if you look into the ratio of deaths, the statistics of death because of the physical damage and because of the blood loss, the ratio is 20:80. That is, for the physical damage, around 20% of the people, they die at the spot. And because of the shortage of blood, because of the loss of blood, around 80% of the death takes place. Now, is there an option wherein we can get the patient, and then, when we rush it into a casualty or into an emergency, there are a lot of assays, there are a lot of, tests which has to be done before the blood is actually transfused into the patient. Now, example, I need to look into his blood type. I need to look into what exactly is his RH factor, which is RH positive or RH negative. We need to also look into the various kinds of infected infectious microorganisms, which can actually, worsen the condition, and all these kind of criteria. Now, under this point, can I design, this brings to a very important component or a very important question, can I design something or any kind of a liquid which can actually save the patient? Now, when I say actually save the patient, I am not talking about the entire antigen antibody or the entire concoction of the blood. I just want the oxygen keep running into my system. So I want to keep the oxygen which is circulating in my system, and it should not hamper any of my vital organs. So this comes to a point wherein most of the patients, they die because of the lack of or the loss of blood rather than the physical damage. Now, when we bring people into casualty or into emergency, transfusing blood is not that easy because there are certain emotions and chemical and biochemical factors which are influencing the blood transfusion. Now, let us try to see what are these factors. Now, the first factor is the orthodox factor or the theological factor. In certain religion, it has been said that the blood cannot be transfused from one person to other person. So that becomes a major criteria for inhibition of blood transfusion. The second thing is I need to know who is actually donating blood and whether the blood has been donated with the willingness of the donor. Now, the donor should be following all kinds of rules and regulations which have been put up by WHO. And when it has been received, the hospital or the blood bank should make sure that none of the blood-borne diseases are being transmitted through this blood. Very importantly, HIV and hepatitis B. Now, the other important criteria is the hepatocyte requires around 24 hours to reach the full oxygen transport capacity, and hence this also plays a very important factor. Apart from that, we need to check upon whether the whole blood was stored in proper refrigeration of between 2 to 6 degrees. And remember, the human blood has a shelf life of not more than 42 days. Now, the blood should not be older than 42 days because that will lead into expiry. And also, very, very important, I need to look into the blood type incompatibility. So, all these factors should be taken care when the blood is being transfused. Now, what are the ideal features of artificial blood or its substituents? Now, why should I do this? Why should I synthesize artificial blood? Now, when I look into the contrast of the real blood or natural blood, artificial blood could be proved to be cost efficient. It can have longer shelf life because in the previous slide we spoke about the 42 hours of shelf life of human blood. So if I'm able to increase it for n amount of time, then this would be again a major advantage. Then storage of blood under a given temperature again is a very, very important criteria. And then the side effects of the epitopic or paratopic side effects which comes with the blood grouping, okay, that can be minimized with artificial blood. And then artificial blood can be made sure that it is entirely pathogenic free, and then it belongs to the universal compatibility. This is a huge boon. Now, what do you mean by universal compatibility? Now, as I told you, Karl Landsteiner was able to classify human blood into A, B and O type. Now, when I take a patient to the hospital, the patient has to be tested for what type of blood grouping he belongs to. Now, under this condition, this takes up a lot of time. Now, by any point of time, if I am able to directly transfuse blood without any testing, okay, which can act as a biological fluid, okay, in his system, which can do only one function, and that function is to transport oxygen. If that is the case, then it is a great hit wherein we can make blood as universal compatibility. And then volume expansion. I can expand the volume because not everybody requires 1 liter or 2 liters of blood. Now, depending on the injury, we can have the volume expansion. Then the oxygen carrying capacity could also be increased or decreased depending on the chemical or synthesized blood. And then the increased availability of the artificial blood would be much more, so that nobody in the entire world or the entire universe will die because of the shortage of blood or because of the loss of blood. Now, these are the major advantages or the characteristic feature that why should I actually synthesize blood. Now, if this is the case, then we will talk about how can I synthesize artificial blood. Many researchers from a very long time, they have put their time, energy, money into designing something which are called as the compounds which can carry oxygen. And one such mega hit molecule are called as perfluorocarbons. They are also called as PFCs. Okay, so these perfluorocarbons are very much efficient in carrying oxygen because of their chemical nature. Now, perfluorocarbons are of low molecular weight, and many times they come in either in a cyclic form or they come under a linear structure, wherein they have a substituted, the carbon chain has a substituted fluorine atom, which leads to the total chemical inertness and complete lack of metabolism in vivo. These are the two important components what we need to remember. So the in vivo metabolism with this particular molecule is absolutely zero, so there is no toxicity, and very importantly, the molecule itself will act as inert. Now, with this property, the perfluorocarbons are not hydrosoluble, again another advantage, and hence has to be used in form of emulsions. And these emulsions of perfluorocarbon are called as PFCs, which are called as perfluorocarbon emulsions. Now, to look into the particle size, the PFC particles are about 1.2 microns in diameter, that is 1/40th time of an RBC cell, which can actually help in adhering to the core of perfluorocarbon with a small lecithin phospholipid as a coat, and this will in fact act as a real molecule which can transport oxygen. Now for this, to make you understand, let us have a small illustration. The illustration is, you have an RBC cell which is around 8 micrometers in its diameter. Then you purify hemoglobin, and with that, if you can have pasteurization and nanofiltration, you have a molecule which is called as HbCOs. HbCOs are hemoglobin carrying molecule, oxygen molecules, or hemoglobin like carrying oxygen molecules, which has a major advantage of no pathogens and there is absolutely no blood typing required. Now, when these HbCOs are being encapsulated with the liposomes, and then with the gas exchange, they're able to actually transfer oxygen, and then the other component is they're able to circulate oxygen to all parts of your body, wherein this size increases from the 8 micrometer to 250 nanometer. So here, the size actually decreases. Now, when the size decreases, the oxygen carrying capacity of these HbCOs actually increases. Now, very recently, this is the news from the last week. Very recently, the Australian academicians, what they have done is they have come up with a mind-blowing technology which has a proof of concept, wherein they are trying to utilize robotics, that is nanorobotics, into the blood, so that the nanorobots are being efficient enough to fight against any kind of blood related disorders. Now, this would be also an add-on in an artificial blood, if this also can add on as a nano or a DNA nanobot or a protein nanobot, which can actually increase the payload in the circulatory system. It would be a great hit. And many times, rather than taking up a vaccine, maybe a technology would be developed so that the nanobots could act as one of the major COP mechanisms, or the major defensive molecules which can nullify any kind of pathogen. Now with this, if I want to conclude, the conclusion for this today's lecture could be, the experiments, whatever we have got, the theories, whatever we spoke, all at this point of time could look very crude. But however, thanks to all those researchers who actually put up their, lot of time, thoughts in establishing the concepts for which we have reached a safe standards today. So the only thing that will never change throughout the entire, the entire evolution or entire understanding of blood physiology is the importance of blood. Remember, yesterday, today, or tomorrow, blood will be the biological fluid for life, and the importance of blood will remain as as prominent as it is today, if we are not able to come up with some innovative and creative solutions. So at the end, I just want to quote William Harvey. As William Harvey always pointed out, any discussion would still end up with blood with William Harvey. William Harvey rightly pointed out, saying that all we know about blood is still infinitely less than all that remains unknown. Okay, this is a great sentence. It is not just applicable for blood physiology, this is applicable for our knowledge. So thanks to the great man who actually explained the closed system of circulation. But with this thoughts, if we are able to come up with synthetic blood, and which has huge advantages, but however, a lot of research, a lot of funding has to be put in, so that we come up with something which is called as artificial blood, or which is called as bio-ink. Okay, thank you very much for your patience, and hope any of you as my viewers, you will be able to come up with the new concept and make this concept bring into reality, so that a boom can be created and nobody dies with blood loss. Thank you very much for watching it. And for any such kind of videos, please let us know so that we can create a new content for you, so that the creativity or innovation in you could be kindled. Thank you very much. See you in the next video.